Good afternoon. I'm here to talk to you about life, about biology and the intricacy and complexity that surrounds us in the natural world. The feathers of a peacock have symmetry and brilliance of color. The wing of a butterfly has both brightness of color as well as structure and strength to allow it to move. The nautilus shell is biology in its most artistic form with a regular pattern of color and structure and texture. And if you look closely at a leaf, you start to see some more of this complexity that we find in biology. If we look a little bit closer, we see that some of these complexities become somewhat simplified, and more of the details come into view. What from a distance appears to be a sea of solid green is actually revealed to be a pattern of individual plant cells, most of which are actually vacant space, with tiny organelles called chloroplasts that actually give this green color that we see. And if we could go even deeper at a smaller level, like Lydia did earlier today, you would see that chloroplast is composed of even smaller structures that give it both its color and its function. When we like to go in closer and we envision what's happening inside of a cell, this is what we see. We see this network of reactions, biological pathways that actually lead to that structure and function that you see in biology all around you every day. Each of these individual dots represents a molecule, and the lines between those dots are chemical transformations that actually allows for all the reactions that we've talked about that gives what we see in biology around us. The significance of that network is that it means we can take really simple inputs, things like a sugar glucose, maybe some oxygen and some other nutrients, and allow that network to be transversed in a way that we get a number of different compounds that can be made. Perhaps molecules that can be used to treat fungal infections, or to lower cholesterol, or to be precursors to a treatment for malaria. Ones that can be used for food flavoring, or even as a platform chemical from which other materials that we use every day can be constructed. If you look at a molecule like this, it's actually made by a plant, by a small tree. And a lot of the compounds we find made in plants can't actually be scaled up. We can't put a plant into a chemical reactor and have it be optimized and produce its compounds at the levels that we would need to satisfy a demand for treatment. But fortunately for us, the blueprint that leads to the construction of that molecule, and really any molecule that we find in nature, is all found in that DNA. Those lines, those connections between those points are related to specific proteins called enzymes. Each of those enzymes can be correlated to a string of DNA called a gene. And we can take those genes from anywhere in nature and put them into something much simpler. We prefer to use E. coli and use that simple system to take those same inputs, sugars and oxygen and nutrients, and have a wide variety of chemical compounds made as a result. And now this small cell is something that we can scale up by fermentation to produce the molecules that we want. In this case, we've been able to make a compound called glucaric acid. This is actually a molecule that we're looking at as a platform to be able to make a new kind of polymeric material that gives us new function. And this is actually made in nature. It's made by plants as well as by animals. But it turns out that the way that nature does this is not very efficient. And I can tell you the way nature chooses to do this would actually more likely instill fear and trepidation in a graduate student than inspire them to do something great. Instead, we could take three genes, just three genes, from three distinct organisms, from a yeast, a mouse, and a bacterium, put them together in E. coli and get them to make this compound. Now, at this point, I always think there's a joke in here, and it starts like this. A yeast, a mouse, and a bacterium walk into a bar. And then I get stuck, and I can't think of any other punchline than just a different artistic rendition of glucaric acid. So let's get back to our story. We've also been able to use these same principles to make this compound called 3-hydroxy-gamma-butyrolactone and a family of related structural compounds, now taking 4 genes from 3 different organisms. And what made this different was that this was a molecule that we couldn't find in nature when we started this work. That means we had to ask those enzymes to do their chemistry on molecules that they had never seen before. And fortunately for us, biology allows us that flexibility in order to make this happen. So we could take that and extend it even further and make a molecule called 4-methylpentanol. This we've predicted to have much better biofuel properties than existing molecules. And you can see more complexity, 10 genes now from 8 different organisms, also making a compound that we have not found anywhere in nature. This lets us know that we really can push the boundaries and use biology to make a lot of chemical compounds. As we do this, we want new chemical functionalities. We want our processes to be economical so that they can compete. And all the while, we want to actually aim for increased sustainability by displacing fossil feedstocks and using renewable sources in between. And we want to reduce the environmental footprint by using less energy-intensive processes. We've been able to apply other concepts of chemical engineering to biology. In chemical engineering, we talk about pipes and flows, where we can actually use valves to direct the way the material flows from one place to another. We've extended this concept to biology and actually been able to construct systems to allow us to direct how metabolites flow inside of a cell. So what I have shown here is a zoomed in view of part of that network that I showed before, where you can see glucose can come in and go in a number of different places. We can create our valves by actually controlling how much of these enzymes are present. And in some cases, we have ones that we can permanently close. We have others that are naturally restricted. And so then we can use engineering to design a system where if we allow this valve to be open, this glucose can go into normal metabolism, the cells grow, they produce energy, and do what they've always done. If we close this valve, it actually causes the material to back up. And now we can make a new compound, glucaric acid. This technology actually allows us to do this much more efficiently than we could otherwise. I actually started my talk with pictures of the diversity and the beauty of nature. And if I ask you to think about chemistry, this is probably more of what you had in mind. And this is great because it shows that we're really dependent upon these large-scale processes to give us the critical materials we need for our everyday lives. But it stops short of really showing us the significance and the elegance of the underlying technology. I hope that in the past few minutes you've been able to understand why for our lab, we think this is a much better view of chemistry, and it really conveys how we see the future, that is, using biology for chemistry's sake. Thank you very much.